Do I have a mic? Am I there yet? Yes? Wonderful. All right, congratulations all of you on your endurance. I'm really impressed to see so many of you still here. Thank you so much, Jamie and the XPRIZE team, for inviting me to join this meeting. And so my job today is to talk to you about a pilot study which we recently undertook at the Buck Institute for Research on Aging over in California with the hope that some of the thought processes that went into study design and some of the lessons that we learned might be useful for those of you who are thinking about putting together a team and what a proof of concept study might look like. So the timeline for this study was that we started thinking about it in 2021. We successfully got funding and IRB approval in 2022 and did a lot of the data collection in 2023. And so all of that is to say was that this study wasn't designed for the XPRIZE, but a lot of the problems and issues that XPRIZE teams will be thinking about were the same ones that we were facing. So we had never recruited participants on site at the Buck, and so we needed to establish that recruitment pipeline and also reliably being able to measure our endpoints of interest. Secondly, our candidate intervention had little to no safety and tolerance data or PK or PD data in older adults. So this was a big gap that we wanted to address before we could move on to a larger study in an older adult population. And really, finally, there was a big gap. We needed to gather data that would allow us to better select population and endpoints for our follow-on studies. So the intervention itself is based on ketone biology, and I'm not going to go too deep into the background here, but I will say that ketones are small molecules that are produced from fat in the liver, and when they circulate in the blood, they can be used as an energy source, but in the last few years, it's been increasingly appreciated that ketones have a vast number of non-energy signaling effects, and that together, these energy and signaling functions intersect with multiple of those hallmarks of aging, which you've been seeing all day. Also, further support for a role of ketones in Lifespan and Healthspan is that dietary interventions, again, that we've heard about today, like caloric restriction and the ketogenic diet, those can also extend lifespan and healthspan. So this is sort of the basis for why we're interested in ketones. Our geriatric syndrome of interest is frailty. It's a multifaceted disease, multifactorial in its causation, and it has a very big, important clinical impact. And importantly, there are multiple areas of ketone biology that intersect with the mechanistic hallmarks of frailty. And so in order to induce ketosis without any dietary changes, so you can induce ketosis without lowering carbohydrate or caloric intake, we use a suite of molecules that we call ketone esters, and you can see a structure up there on screen. These are molecules, when you consume them within 30 to 60 minutes, it might be like you'd been fasting for a week. So they're very rapid and repeatable ways to induce ketosis. And from a scientific perspective, it's interesting as it really allows us to isolate the effects of ketones from any other changes in the diet, so as I mentioned. But again, as I said at the outset, before we could move ketone esters into an older adult population, there were some really key gaps that needed to be addressed to give us confidence. Almost all of the studies that had been done were in younger adults. A lot of them were in athletes and acute studies. There was nothing longer than 28 days and nothing in a population over the age of 65. And whilst, as I've alluded, there is a lot of preclinical data that links ketone biology with aging biology, that had never really made the jump through into a clinical trial. So we were really interested to see whether we could recapitulate some of the promising preclinical data in a human and translational model. So along with my mentor, Dr. John Newman, who deserves the biggest shout out in the world, and I'm sure he wishes he could be here too, we undertook this randomized and double-blinded, placebo-controlled pilot study of 30 healthy older adults. I would say here, a theme that sort of was mentioned by an earlier speaker was these were not completely healthy. We did have people with stable chronic diseases because it's very, very difficult when you start having a very tight inclusion criteria for these studies. So some of the elements that we addressed were our older adult specific kinetics. So both before the intervention and after the intervention, we tested the effects of 2 ketone ester formulations, a beverage and a powder, on blood ketone concentrations in our older adult population. Then at baseline, we not only collected biospecimens, as you'd expect, so we collected plasma, We collected PBMCs, stool, and urine. That was to give us hopefully make those mechanistic links between aging and biology and ketone biology. We undertook physical function testing, cognitive function testing, and quality of life testing, and a number of the measures that we used were discussed earlier today by Dr. Cummings and some of the other speakers. So our physical function measures included the 6-minute walk test and leg press strength. Our cognitive function outcomes included digit symbol substitution and trail making tasks. And our quality of life functional endpoints focused on sleep, mood, and fatigue. And so we were using these very standardized measures that could be used in any study of any intervention to look and make links between ketones and aging. So after the baseline testing, all the subjects were randomized to receive either ketone ester or placebo. We had a 2-week dose escalation period built in because we felt that that would improve our tolerance. And I would like to highlight that tolerance and safety were our primary outcomes for this. So we weren't focused on any of that fancy biomarker stuff or the physical cognitive function. We really just wanted to get a good read on if this was a feasible intervention in an older adult population. In the spirit of that, we had a safety check at week 4, and then everyone came back again at week 12, and we repeated all of the outcome measures that we had taken at baseline, and we were looking for within individual changes across the 12 weeks of the study. And the study was a really big success. Our primary outcome, which was chosen based on the symptoms that we'd seen in younger adults taking ketone esters, and also just the expectation that in older adults we might be getting sporadic symptoms day to day, was the proportion of subjects experiencing a moderate or severe dizziness, headache, or nausea on more than one day after our 2-week dose escalation period. And you can see here that there was not a difference in subjects meeting that primary endpoint between our 2 study groups. Overall, our side effects were low and not different between our 2 study groups, and we didn't have any serious adverse events. Although we had 6 subjects not completing, only 2 of those were in the ketone group compared with 4 in the placebo group. When it came to safety, we didn't see any changes in vital signs, and we had picked our safety labs based on an expected interaction with ketone biology, so lipids, liver function, and we didn't see any changes in those either, so that was all very reassuring. Our kinetics data was similarly illuminating, and you can see here on the left, firstly, in red, the blood ketone responses to 12.5 grams of the ketone ester, and in blue, the blood ketone responses to 25 grams of ketone ester, so we saw that it was nicely dose responsive. If you look on the right, you're seeing that same data in blue from our older adult pilot where our median age was 76. Plotted alongside with a slightly younger cohort, middle-aged cohort with a median age of 51, which we had undertaken earlier using an almost identical study protocol, dosing, and all of those things. And it did show us that we're likely to be having an age-specific metabolism of these ketone ester products that needs to be accounted for in future study design. In terms of our kind of exciting secondary and exploratory endpoints, a lot of that analysis is still ongoing. We're very excited to start seeing a signal in our proteomics data linking ketone biology with cellular senescence, and also in our microbiome data as well, where we're seeing reduced inflammatory markers with our ketone intervention. We did not see any changes in our physical, cognitive, or quality of life outcomes, but honestly, that's not really surprising in a very small study where the population was already quite healthy and high functioning and the relatively limited duration. But it did help us. All of the information that we gathered was useful as we thought about designing our follow-on study. So our follow-on study has been successfully funded by the NIH. It's an R01, and the data we got in our pilot's given us confidence to make a number of changes that you can see here. So firstly, we've expanded the sample size a lot. We're now going to be recruiting 180 individuals across 3 sites across the US, and that'll help us increase the diversity of our sample as well. Based on our great safety, tolerance, and adherence, we feel comfortable increasing our dosing from 25 grams once daily to 25 grams twice daily, and this hopefully will give us a better chance of detecting effects. And similarly, increasing our intervention duration from 12 to 20 weeks. For this next follow-on study, we're going to be including patients who have a slight functional deficit at baseline. So a slightly slower gait speed. We're asking people if they feel like they're starting slowing down, if they have a bit of difficulty walking 1 or 2 blocks, and those will be our participants. And that is because we feel like we're more likely to both effect and detect an effect of our intervention in this population. We'll be collecting additional biospecimens, so we'll be collecting muscle biopsies for multi-omic and potentially even mitochondrial functional analysis, and we'll also be continuing to collect PBMCs, so white blood cells, and doing some deep immunophenotyping as well. But I did want to, at the last, draw your attention to our choice of primary outcome, which was kind of chosen after some discussion with the community here. We have decided to go for a composite outcome that is a scale from vigorous to frail. We think that composite outcomes are interesting for gerotherapeutics because, as George Kuchel really nicely showed earlier, gerotherapeutics, they can be having modest effects against multiple mechanisms and multiple endpoints that taken together are clinically significant, but may not be statistically significant by themselves. So that's why we decided to move to a composite outcome. The composite that we will be using was developed by the team behind the SOMA study, which you've heard discussed earlier. And this is a very large and high-quality cohort. And using the data that they've already collected, they were able to build this composite with endpoints that are very predictive of progression from vigorous to frail. And it's also a continuous measure, which gives us a lot more power to kind of discriminate between smaller changes, especially when people are more functional. And so the elements that are going to be included in the composite you can see here also directly relate to multiple elements of the very well clinically established Fried frailty phenotype. So we're excited to implement this in our ongoing study. So that's it from me. Hopefully you've seen how a relatively small study. I would say that the donation that funded this study was only $250,000, so it's very, very similar to the amount of money that the 40 semifinalists will receive. How a study like this can de-risk things, provide a lot of very, very important information as you move on to design bigger studies and potentially even some fun effects that hint at mechanism and clinical efficacy as well if you're lucky. So I'd like to wish all the teams a really good luck and close by again thanking Dr. John Newman, Eric Verdin, who is going to be here later this week. I got to shout him out. He's my boss. Our fantastic clinical team. It takes an army to run this kind of study, and we were starting from nothing, so we all learned a lot. And finally, as I said, the study was funded by a philanthropic donation from an individual, Dr. James Johnson, along with the Buck Institute Impact Circle. So we are very grateful for them making this work happen. Thank you all.